Hello friends, how are you doing? And I hope you all are doing good and studying well, concentrating well, revising stuffs at regular intervals and concentrating on your hard work. Yes, so we will now today discuss about the NEET pattern exercise, respiration in plants. And let us go to the first question. Whenever you are doing questions like this, you always you don't hurry, don't rush up looking at the question. Just don't try to jump up to answers. Everything everything. If you have studied properly, most 90% of the things you will know, but only if you concentrate, and if you do not get restless, things will be very easy, believe me. Which of the following, even at this level when we are teaching, we have done this like n number of times, but even looking at that, if we try to jump questions, we will make mistakes. We have to read the question, stay calm and positive, and things will flow. So which of the metabolites is common to respiration mediated breakdown of fat, carbohydrate and proteins? So this is talking about something, okay, this is talking about the glycolysis, right? So glycolysis, right? So which of the following, actually it's not completely talking about the glycolysis, it is telling that which of these is common to respiration mediated breakdown of fats, carbohydrates and proteins, right? So all of these, whether we talk about fats, whether we talk about carbohydrate, or we talk about the proteins, what we will see that they will undergo glycolysis. And what happens in glycolysis, we know that glucose forms glucose 6-phosphate, right, glucose 6-phosphate, and this forms further fructose 6-phosphate, and then this will form what, fructose 1,6-biphosphate. We have done all that, fructose 1,6-biphosphate, right, fructose 1,6-biphosphate, right, all that. So the end product of glycolysis will become what? Pyruvic acid. Hence end product of glycolysis we will see is 2 molecules of pyruvic acid, okay, 2 molecules of pyruvic acid, okay. So hence the end product of glycolysis, is 2 molecules of pyruvic acid. And what will we see? What will we see further? Further what will we see? That there will be oxidative decarboxylation, and in that oxidative decarboxylation, this pyruvic acid, okay. So we can write that down. In oxidative decarboxylation, right, in oxidative decarboxylation, the pyruvic acid gets transformed into acetyl, okay, into acetyl coenzyme A, into acetyl coenzyme A, right. So in oxidative decarboxylation, the pyruvic acid gets transformed into acetyl coenzyme A, and then then this can have any route. okay, it can get into the mitochondria for the Krebs cycle and so on. So, let us now do the options. So, which of the metabolites is common to respiration mediated breakdown of fats, carbohydrate and protein? So, immediately glucose 6-phosphate cannot be the 1-fructose 6,1,6 biphosphate. That was one of the, these were in one of the, in fact even the pyruvic acid, these are all the intermediates of glycolysis. This is the end product of glycolysis. And then we see that the pyruvic acid will move on to the fate acetyl coenzyme A, right, will move on to the fate acetyl coenzyme A, and hence this will be the correct option, right. So, now we will talk about the next question. Okay, we will talk about the next question. The chemiosmotic perk coupling hypothesis of oxidative phosphorylation proposes that adenosine triphosphate ATP is formed because, so it is talking about the formation of ATP, because high energy bonds are formed in the mitochondrial proteins. Does not make any sense. The ADP is pumped out of the matrix into the intermembrane space. ADP is pumped out of the matrix into the intermembrane space. Not make. A proton gradient forms across the inner membrane. Now, this makes sense. Let us see the last one. There is a change in the permeability of the inner mitochondrial membrane towards the ADP. Not actually. So, the third option, the C option, will be the right option, and we will see how is this, right. We have all talked about that the set of electrons, the electron carriers are located, these are the cristae. This is the intermembrane space, okay. So, let us forget about the right, this is the mitochondria, if you think as, right, the mitochondria. And this is the lumen of the mitochondria, right, and the mitochondria is situated where in the size, in the in the cell, okay, in the cell, one of the organelles of the cell, right, and this is the outer membrane and the inner membrane, right, the intermembrane, this is the intermembrane region. Right, why? Because this is the outer membrane and this is the inner membrane. Now, what happens from here, the matrix, what happens, see, well, we know that the electron carriers are located here, electron transporters, electron they will carry, the electron transport chain is located in the inner membrane of the mitochondria, right. So, we see that the electron carriers are situated here, the electron carriers are situated here. Now, what happens, when the electron is being transferred from one electron carrier here to the other one, what happens, utilizing this energy, the proton the increase in pace was to wake you up from sleep, okay, the mitochondria. So, what is happening, as electrons get transferred from one electron carrier to the other, there is the proton ion, these are the proton ion, and these will diffuse from this matrix right into the lumen, right. There is now a proton, there is more of hydrogen ion, more of the positive charge inside as compared to outside, that is a matrix, and hence we see that yes, okay, I have whatever I have written. So, lumen, and hence we see that the inner gets more positive than the outside, the inner gets more positive than the outside, right. Then what will happen, the inner gets more positive than the outside. Now, this hydrogen ion has to come out again back into the matrix, and how does it do? There is something called as the F0 F1 ATPase enzyme, right, the adipase enzyme, and this adipase enzyme, okay, this hydrogen ion now tries to come out through this. Now as this hydrogen proton ion is coming out, what happens, you know, the energy of this proton is being utilized by the adipase enzyme, okay, adipase enzyme, and at this region, at the F0 region, sorry, F1 region, we see that ADP gets phosphorylated to with phosphate to form the ADP. This is what we are talking about. The chemiosmotic coupling hypothesis of oxidative phosphorylation proposes that the ATP is formed because the proton gradient forms across the inner membrane. Proton was more in the matrix before, less here, but you know, inside it has come. Also, you know, also one of the reason why proton is more inside is there is photolysis of water. There is a photolysis of water inside. And you know, this photolysis of water is giving what? Is giving the H plus ion plus OH minus ion. So this H plus is also contributing to the increase in proton ion inside the lumen, and as a result the proton gradient increases inside the lumen, which is abnormal, which is not, and hence there would be no stability, so it has to move out. Yes, I hope you understood this. Great. Now we move on to the next question. All enzymes of the tricarboxylic acid cycle are located in the mitochondrial matrix, except one which is located in the inner mitochondrial membrane in eukaryotes and in cytosol in the prokaryote. Right. Okay. So all the enzymes of TCA cycle are located in the mitochondrial matrix, except one which is located in the inner mitochondrial membrane, okay, which is located in the inner mitochondrial membrane in the eukaryotes and in cytosol in the prokaryote, and you know, it tells which is this enzyme. Now this enzyme is the succinate dehydrogenase. The enzyme succinate dehydrogenase, okay. The enzyme succinate dehydrogenase is located in, where is selling, in the inner mitochondrial membrane, in the inner mitochondrial membrane. In the inner mitochondrial membrane, right, and it converts the succinate to fumarate, okay, and it converts the succinate to fumarate. Right. So the enzyme succinate dehydrogenase is located in the inner mitochondrial membrane, and it converts the succinate to fumarate. Okay. So the answer for this will be succinate dehydrogenase. Succinate dehydrogenase. Okay. We move on to the next question. Site of respiration in bacteria. Right. Site of the respiration in bacteria. The inner membrane of the bacteria, the inner membrane, okay, the membrane of the bacteria shows infolding, right? the membrane of bacteria shows infolding right. The membrane of bacteria shows infolding called as mesosomes, okay, and these mesosomes have enzymes for respiration, okay, and these mesosomes have enzymes for respiration, right. So the membrane of the bacteria shows infolding called as mesosomes, and these mesosomes have enzymes for the respiration. So site of respiration in bacteria is the mesosomes. This is the correct option, right. Now we move on to the next question. Oxidative phosphorylation involves simultaneous oxidation and phosphorylation to finally form what? So, oxidative phosphorylation itself means, there will be an addition of ADP, of phosphate to ADP. Okay, the word phosphorylation itself tells you that, the phosphorylation, that is addition of phosphate group to ADP to form, hence, if a phosphate group is added to ADP, it will definitely form ATP. So, the word itself here says that yes, the answer will be this. Now, what is happening, in this oxidative phosphorylation, what is happening, there is transfer from the FMN or the NADH to the oxygen, and as a result, what is happening, the ADP is getting synthesized. As a result, the ADP is getting synthesized. So, the correct option or the answer will be ATP, right. The respiratory quotient. Okay. So, let us see what is the respiratory quotient, and it is asking for the fatty acid. Respiratory quotient RQ, okay. So, respiratory quotient RQ for a substance, will range, right. So, respiratory quotient RQ for a substance will range between the amount of carbon dioxide evolved, to the amount of oxygen which is breathed in, which is being utilized, okay. So, respiratory quotient for a substance will range between the amount of carbon dioxide evolved to the amount of oxygen to the amount of oxygen which is being utilized, and hence RQ will become, respiratory quotient will become carbon dioxide evolved to oxygen consumed. The respiratory quotient for, so the RQ can vary. The RQ can be less than 1, it can be 0, it can be greater than 1, it can be even infinity. All these can be the range of the respiratory quotient. Also we will see, in case of the fatty acid, in case of fatty acid we will see that the carbon dioxide evolved is much less as compared to the oxygen consumed, because it has to undergo the oxidation, right. So the carbon dioxide evolved is much less as compared to the oxygen consumed, and hence the RQ for fatty acid will be somewhere around, RQ for fatty acid will be somewhere around 0.7, okay, somewhere around 0.7, and hence it is yes, less than 1. So the respiratory quotient of, that is RQ for fatty acid, is less than 1, right. Now we move on to the next question. ATP is injected in cyanide poisoning because it is necessary for cellular functions, right, necessary for sodium potassium pump. Sodium potassium pump operates the cell membrane and ATP breaks down the cyanide. Now what is happening in cyanide poisoning? There is a stoppage, or there is a ceasing, there is a break in the electron flow, okay, from cytochrome B to cytochrome C1, right. So in cyanide poisoning, the electrons do not flow, okay, the electrons do not flow in the electron carrier. In the electron carrier, this is 7th question, from cytochrome B to cytochrome C1 to cytochrome C1. Okay. So hence we all know that ATP, hence ATP will not be synthesized, and inert ATP is very, very important for all the cellular functions of the body. Now you must be getting an option as to getting a hint as to what must be the option for this. So in cyanide poisoning, the electrons do not flow in the electron carrier from cytochrome B to cytochrome C1, hence ATP also not synthesized, right. Hence ATP also not synthesized, and what we will see further, that yes, ATP has to be injected thus from outside for the cellular functions. Hence ATP is injected, hence ATP is injected, which is very necessary, to maintain the cellular functions, to maintain the cellular functions. Hence ATP is injected, which is very necessary to maintain the cellular functions, right. So, necessary for the cellular functions, ATP. We move on to the next question. At a temperature above 35 degrees centigrade, okay, at a temperature above 35 degrees centigrade, the rate of photosynthesis will decline earlier than the rate of respiration. The rate of respiration will decline earlier than the rate of photosynthesis. There is no fixed pattern and both decline simultaneously. Okay, wait, I hope so. So, what is happening, first we will see the optimum rate for, photosynthesis. Okay, I think this is, a favorite sign for one of the actors, right? Kamosh, Shatrughan Sinha. Okay, the optimum temperature for photosynthesis for C3 plants is what? 10 to 20 degrees centigrade, and the optimum temperature for photosynthesis for C4 plants is 20 to 35 degrees centigrade, right. 10 to 20 degrees centigrade and 20 to 35 degrees centigrade, and the optimum temperature for respiration will be somewhere, for respiration will be the optimum temperature for respiration will be how much? It will be somewhere around 25 to 35 degree centigrade, right. So, what do we see? The optimum temperature for respiration is more as compared to these two, but I mean, and C3 plants are, we are a major concern. So, hence when the temperature will rise, the rate of photosynthesis will decline earlier, because as the temperature is increasing over 35, yet that temperature is near to the optimum temperature for respiration, but it is going away from the photosynthesis, and hence the rate of photosynthesis will decline earlier. Yes, I mean, you can just than the rate of respiration. Got it? This is how you got to understand. Okay, we move on to the next question. Out of 36 ATP molecules produced per glucose molecule during respiration, how many are produced before and how many are produced later on? Right. So, we all know that out of the 36 ATP molecules produced per glucose molecule, 2 are produced outside, that is in the cytosol during glycolysis, and 34 during the respiratory chain. 2 are produced outside the mitochondria and 34 inside the mitochondria. Wrong. 2 during glycolysis and 34, not 34 entire is not produced during glycolysis Krebs cycle. So, the correct answer will be 2 are produced outside the glycolysis and 34 during the respiratory chain, right. Now, we move on to the next question, right. The NADP plus is reduced to NADPH in what? What we will see that the NADP plus is reduced to the NADPH in the hexose monophosphate pathway or the pentose phosphate pathway, what we call this as, you know, the HMP. So friends, I hope, you know, this is clear and everything is fine and perfect and absolute. Okay, you're following things, and you know, any doubt you can always come back to me. Okay, the time we meet again, a very goodbye from this side. Thank you.